When we discussed DNA replication earlier in the quarter, we talked about DNTPs, and we said these were nucleotides that are the building blocks of DNA. And we said that if we're talking about DNA, there's going to be one of four bases, A, T, G, and C, a 5-carbon sugar, and we're going to have 3 phosphate groups coming off the top. Now, when this DNTP gets joined on to the growing polynucleotide strand, 2 of the phosphate groups were removed, so there was 1 phosphate group between the sugar and phosphate, the next sugar, a phosphate, and so on. So that's a DNTP. Now, the purpose of DNA sequencing is to determine the order of A's, T's, G's, and C's on a specific strand of DNA. And the technique used to determine that is again called DNA sequencing. DNA sequencing relies on a variation of a DNTP called a ddNTP. So DNT stands for deoxynucleoside triphosphate. ddNT stands for dideoxynucleoside triphosphate. Now, chemically, what's the difference between deoxy and dideoxy? You'll remember that on the 2' carbon we have an H, on the 3' carbon we have an OH. So that's a deoxynucleoside. There's a 3' OH group. On the ddNTP, in both the second and the third positions, there is no oxygen. So that's said to be a dideoxy. In other words, you've taken away 2 oxygens. Now, why is that important? The answer is, remember, to extend the chain, you have to have an OH group in the third position to add the next nucleotide onto. If a ddNTP is inserted as DNA is replicating, then it will terminate DNA replication, meaning that because there's no oxygen on this 3' carbon, another nucleotide would not be able to join. And so this dideoxynucleotide is very helpful for DNA sequencing, and I'm going to show you how it works in more detail on a later figure.